Hi everyone. In the previous part, that is part 1, we have learned about direct microscopic methods to measure cell numbers. Now, in this part, we are going to discuss about indirect methods of measurement of cell numbers. Now, let's have a quick review of methods of measurement of microbial growth. So we know that we are having three methods. The first one is going to the measurement of cell numbers, in which we have the direct cell count and the indirect method. And the second one is measurement of cell mass. And the third one is going to be the measurement of cell activity. So coming to the first type, measurement of the cell numbers. So we have two methods here. One is going to be the direct cell count or total count that we discussed in the part 1. And the second type is the viable count or indirect method that we are going to discuss in this part. So where the measurement of cell numbers by indirect method is going to possess the two techniques. One is going to be the plating method and the second one is going to be the membrane filtration method. So in the indirect methods of measurement of cell, these two were included, that is plating method as well as the membrane filtration method. So let's see these things in detail now. Coming to the plate counting method or technique. So here in the plate counting method, we are going to detect mainly the viable cells. So viable cells or the cells those are able to form the colonies on an appropriate solid nutrient medium. The two routinely used methods of this plate counting methods are, one is the spread plating, another one is going to be the pour plating. So prior to plating, it is usually necessary to prepare a serial diluent series in a sterile medium for concentrated cell suspension. Alternatively, for samples with low numbers, as in water analysis, the concentration step is required. So here in the plate count method, we are supposed to inoculate the plate with a sample and count the number of colonies, and each colony is going to represent from a single bacterium. That means each colony is going to be arised from a single bacterium. How many number of colonies are there? It is directly proportional to the those many number of individual cells or bacterial cells. So here you can see the pour plate method and the spread plate method. What is happening in the pour plate method? So first of all, we have to take the bacterial dilution, and in the pour plate method, you have to take a plate and in which you have to put your sample first. You have to inoculate, and then on which you are supposed to add the melted nutrient agar, and just you have to swirl to mix the medium as well as the bacterial dilution or suspension. Once they were mixed, they were incubated for 24 hours. So after the 24 hours, we are able to see the growth of the colonies in as well as on the solidified medium. And then we will count, and which is directly proportional to the number of bacterial cells. This is the pour plate technique. Then coming to the spread plate method. So here in the spread plate method, on the nutrient agar medium, 0.1 ml of the sample is going to be plated, and that is going to be spreaded with a spreader over the surface evenly. So after that, again this plate was incubated, and after the incubation we are able to see the colonies growth only on the surface medium, not in or under surfaces. So obviously you are going to count the number of the colonies, which is directly proportional to the number of cells. So that's how we are going to have the two things. One is the pour plate method and the second one is going to be the spread plate method. Then coming to the advantages and the disadvantages of the pour plate method. So we are going to measure the viable cells by using this method, and this method is very simple and useful and even sensitive method. Then coming to the disadvantages, the main disadvantages it is going to have in the taking of 24 hours or more for visible colonies to appear, and we can count only between 25 to 250 colonies are accurate, and the low count could be due to clumps, and if the medium is not having the optimal conditions, obviously the count will be low. And the hot agar that we are using may injure the cells in the pour plate technique, and the count will be less. So for certain count, we are supposed to have the serial dilution to get the appropriate numbers. So these are the few disadvantages regarding the pour plate method as well as the spread plate method that are used for viable counts. Then coming to the serial dilution containing viable cell count, here the viable count is a method of measurement of concentration of live cells in a microbial population or bacterial population. The number of bacteria in a given sample is usually diluted and then plated out on a solid medium in a manner such that we are going to get isolated bacteria form visible isolated colonies. So here you can see, so as we are moving on the concentration, here so much of the concentrated bacteria are grown, and if you see this plate, isolated single bacteria were visible. Now the number of colonies formed can be used as a measure of the number of viable cells in that dilution. So where each colony is going to be considered as a colony forming unit, simply called as CFU. So coming to this figure, if you observe, for convenience, we are going to obtain a colony count for bacteria in any sample. We can check the milk sample, water sample, or anything. Okay, so 1 ml of a well-mixed sample is placed in some sort of 9 ml, or otherwise we can take the 99 ml if it is a milk of a sterile dilute solution. Okay, this dilute solution can be a water or nutrient broth or saline solution. Whatever the thing is, you have to take the 9 ml of it and 1 ml of the virginal inoculum. And this is going to give the result of 1:10. And then now this have to be transferred to the next one where you are going to get the dilution of 1:100. To the petri dish, so whatever the thing is, to the petri dish containing pre-solidified medium, 1 ml of or 0.1 ml, I tell you that one. So here 1 ml of this sample was transferred in and inoculated at a desired temperature. Now like this, from every dilution you are going to plate the sample of about 1 ml in the concerned dilutions. The number of colony forming units is calculated after incubation period is completed. So after incubation period, the plates are going to possess this colonies, and those were going to be calculated. The procedure is repeated for the preparation of further dilutions like 1:1000, 1:10,000, 1:1 lakh, like this, or with subsequent plating till easily countable plates were obtained. That means easily countable colony forming units are obtained. A plate having 3 to 300 colonies is chosen because this range is going to be considered statistically significant. If there are less than 30 colonies on a plate, small errors in dilution technique or the presence of few contaminants will have a drastic effect on the final count. Likewise, if there are more than 300 colonies on the plate, that means like this, then there will be poor isolation and colonies will have grown together. So these two considerations we are going to take as a minimum and the maximum. If the probable number of bacteria per ml, 1 ml in the original sample can be found by multiplying the bacterial colony count by the reciprocal of this dilution and of the volume used. For example, here you can see the number of colonies on plate into the reciprocal of dilution of sample gives rise to the number of bacteria per 1 ml. So if we get the 32 colonies on a plate of at a dilution of 1/10,000 or 1:10,000, then the count is going to be of 32. Here number of colonies is 32, and the reciprocal of the dilution means actually you are going to have the reciprocal. So here 1/10,000 is the value, and the reciprocal of it becomes 10,000 by 1, isn't it? So that's why we will neglect this one. So 32 into 10,000, that gives rise to this much, that is 32 into 10 to the power of 4 per ml in the sample. So these many number of bacteria are going to be present in that concerned dilution. Okay, so this is how you are supposed to calculate. Then moving to this what we call formula and the calculation, accurate calculation. So here is the formula for calculating the concentration of the bacteria is the number of colony forming units per ml of a sample is equal to number of colonies, that means how many number of colonies you got, and the dilution factor of the plate counted. So how much you have calculated, that you are supposed to take. Then this frequently the volumes other than 1 ml are used to inoculate the plate. For example, 0.1 ml is oftenly used. So previously I have explained it as a 1 ml, but what we are going to take the low volume, that is of about 0.1 ml, is oftenly used when surface plating, as larger volumes may not be absorbed by the agar. So for that reason we are taking the 0.1. So plating the 0.1 ml of a given dilution is mathematically identical to the plating a 1 ml of a further dilution called as 1:10 dilution. For this reason, the size of the inoculum is usually incorporated with the dilution factor to give the final dilution. So what is the final dilution here? That is D into I. When 1 ml of 10 to the power of -4 dilution is plated, the final plate dilution is 10 to the power of -4. When 0.1 ml of the same dilution is plated, the final dilution is going to be 10 to the power of -5. So that means here, depending upon the dilution, it is just changing. So here this is going to be considered as the final dilution. So if you are going to take the 1 ml of the sample, the final dilution will be 10 to the power of -4. If the sample is going to be the 0.1 ml, then you are going to add 1 by 10 more, so that becomes the 10 to the power of -5. So this is what you have to remember. So the formula for this CFU per ml is going to be coming colonies on plate divided by D into I. So D indicates the dilution factor, so whatever the dilution factor is, and the I is the volume plated. That means whether you plated 1 ml or 0.1 ml. So this is how you are going to calculate the viable counts. Then the advantages of the viable count is going to be of viable count is very useful in the dairy industry as well as in the food industry for qualitative analysis of milk as well as the spoilage of food products. Then the second one, the number of colonies formed can be used as a measure of number of viable or living cells in that non-dilution. So that's how these are the two main advantages of viable count. So we are also having some disadvantages with this one. That means here by using the viable count we can have this only with the living cells, and the other one is dense solutions are going to be needed to get the accurate colony form counts. So that's how these are the two disadvantages of the viable count. Then moving to the next one, that is membrane filter technique. So here the membrane filter technique is an effective as well as and most widely accepted technique for testing the fluid samples for microbiological contamination. And not only that, this technique involves less preparation than many other traditional methods. And this is one of the few methods that will allow the isolation as well as the enumeration of microorganism. That means we can isolate the microorganism as well as we can count the number of microorganism by using this membrane filter technique. Okay, what we are doing in this technique? In this technique, a thin membrane filter disc consisting of cellulose derivatives are going to be used. And the filter disc retains on its surface all the bacteria from the water surface, that is whatever the water containing bacteria are going to be trapped by this membrane filter. Now the water is going to be filtered through the filter disc in this one. So you can see this is the water, and the water is being filtered through this filter disc, and the disc is then transferred with a sterile forceps on a thin absorbent pad that has previously been saturated with the appropriate medium. Now generally the medium that we are going to use here is endobroth medium. Okay, now the membrane was placed in the Petri plate. Now the Petri plate containing absorbent pad and the filter disc is incubated at 37 degrees centigrade for 18 to 24 hours. Now the medium is going to be diffused through the pores of the filter disc and provide the nutrients to the bacterium that is trapped in this membrane. Now the passage of nutrients through the filter facilitates the growth of organisms on the upper surface of the membrane. This is how they are going to. After the incubation is over, colonies are found to develop upon the filter disc, and the characteristic colonies of different bacteria could now be studied to determine the water potability. So this is what happening here, and this technique is going to be of mainly useful in analyzing the aquatic samples. So here you can see. So this is the overall. So we have taken a filter paper and we have kept in it and in a membrane, that means membrane filter, and we allow the water to flow through it, and now this membrane is going to trap the bacteria that were present in it, and then after the filtration we are removing that membrane filter with a forceps, and that was placed in an endo-agar medium in the Petri plate, and then you are incubated for 18 to 24 hours. After the 24 hours, we are able to observe the colonies developing on the filter disc. So the same thing you can see here what is happening. So this is a hand-drawn figures. So the membrane filter possessing millipores small enough to trap the bacteria. So water sample filtered through the filtrate to trap the bacteria on the filter, and then membrane filter removed and placed in a nutrient agar medium containing Petri plate, and then it was incubated and the typical colonies were formed. So like this you can see the typical colonies on the membrane filter. Then coming to the advantages of membrane filter method. So an effective and accepted technique for testing fluid samples, especially the water like that which were contaminated with the microorganism. And this involves less preparation when compared to the traditional methods like our other methods. And it is one of the few methods that will allow the isolation as well as the enumeration. I told you we can isolate the microorganisms. At the same time, we can count the number of microorganisms by using this membrane filter technique or method. And a large volume of sample can be analyzed at a time, and it is especially used in analyzing the aquatic samples, that is liquid. And various types of microorganisms can be detected by using the selective media in the membrane or in the Petri plate. So example, membrane filter method offers the advantage of isolating discrete colonies of bacteria. When we come to that one, that is MPN, most probable number procedure, only indicates the presence or absence of approximate number of organisms. But here we are going to isolate as well as the counting can be done by using this membrane filter method. So this is all about the indirect methods of measuring the number of bacterial cell or measurement of cell numbers. So in the next part, we are going to discuss about the measurement of cell mass by different techniques. Thank you.